All right, awesome. And speaking of AD, kind of related, the second most common neurodegenerative disorder is Parkinson's, and that's going to be what we're going in today. So yeah, I know Professor Winston has to leave a little bit early today. I think around like 5:30, right? For appointment she has. So this
Mm-hmm.
is what I'm gonna try to do. I'm gonna try to go over the slideshow I made and review kind of the research by five. I wrote 5:17, but yeah, around that time.
Mm-hmm.
So we'll have around a little bit over 10 minutes to have a little discussion time with Professor Winston. And then afterwards, we ourselves can have a little bit more discussion time. And then at 5:50, I want to kind of have like an open talk about maybe suggestions for discussions or because I had a couple ideas on what we could do going forward in regards to organizing this discussion or whatever. So yeah, we'll kind of have a democratic process on that. But for now, let's just get started. And also just a reminder, if you can, please turn on your cameras because facial interaction is very important. Okay. Share screen.
Sorry, I would turn on my camera, but my bandwidth is really low, so it won't let me turn it on.
It's all
I'll
good.
be sure to get fixed. Thank you.
Yeah. Um, you're like it's not an you don't have to turn your camera on. If you can, please do. Sorry, 1 second. Right, show. All right. Um, okay, here we go. So the research we went over this week was a little bit more complex, I think, I feel like personally than last week's. But hopefully, if you had any questions, these slides kind of clears up stuff for you, so you can be more confident during our discussions and ask more questions. And yeah, it should be a good refresher since we're having meetings every 2 weeks. So yeah, our research this week was on graphing quantum dots and how it relates with a-syn that relates with Parkinson's disease. So the misfolded proteins. And before I go into anything, I just want to clear up graphene quantum dots are like it's not specifically for this. It like it graphene quantum dots are like a thing utilized for many different things, but for this research particularly, it's we're investigating the relationship between GQDs and a-syn. So that's the main thing we're looking at today. Here are the table of contents. Because this was a like a differently structured article, um, we're gonna kind of see. I guess I'm gonna kind of go down in a different order, kind of to match the structure of the article itself. And here in the introduction, I kind of wanted to clear that up a little bit. It's in a more unique format where it doesn't have like the methods and then the results and everything outlined. It has like a main body article part, and then have it has another methods section that's goes more in depth to the methods. Um, but yeah, so honestly, what I'm gonna do is just go through each of the main points from the main body article and then just kind of go over that. So before we get into anything, here's some background that I came up with for this research. So a-synuclein aggregates. They're bundles of so these aggregates are basically bundles of misfolded proteins. And misfolded proteins are usually never good news. Normal A-syn proteins are their exact purposes aren't 100% sure, like aren't really known, but they're very common in the brain and they're in the synapses, I think. I'm not sure. But they're very common. They're all throughout your brain and they probably do play a key role in your neurological processes. However, their exact roles are still not known. And these normal proteins are usually they do their job, whatever it may be, in your brain. But if they start misfolding, that's when problems start to occur. And misfolded proteins, these aggregates are very harmful because they start doing neural damage as they kind of progress and get bigger. So these aggregates kind of evolve. First off, the a-synuclein protein misfolds, and when it misfolds, it starts kind of bunching up to form fibrils, and these fibrils then also start bunching up to form Lewy bodies, and like these misfolded proteins are basically aggregating together to become larger and larger, and then that eventually leads to like neural damage and like damage to the cells, which can lead to cell death. And cell death in like the substantia nigra in the within the dopaminergic neurons is what specifically leads to Parkinson's disease. So these asyn protein aggregates are very heavily correlated with Parkinson's disease. And okay, so now let's go on to the findings. Um, the first part was basically talking about how what GQDs do in this context specifically. So to simplify everything, they basically do two things, two key things that we need to know. One is that they prevent the fibrillization of misfolded asyns. So like I said, when these proteins misfold, they tend to fibrilize into, fibrils, but GQDs actually prevented the formation of fibrils. Also, they disaggregate previously formed fibrils. So it's basically it prevents it and it also undoes whatever's been done. And that's why GQD is such a great potential therapy for Parkinson's because it can stop it in two different places, before the fibrillation and even after the fibrillation. Um, and then this is a little bit onto the methodology of how the researchers localized and like kind of visualized how the GQDs and the aggregates interacted. One thing they used was biotin hooks. So biotin hooks were essentially just kind of imagine it as a Velcro where the a, a thing known as biotin was attached to the GQDs. And then biotin has a very special binding capability, I guess, with streptavidin, another type of just molecule. And this streptavidin was attached to gold. So basically, what would happen would be that the GQDs would attach to the GQD biotins would attach to streptavidin gold. So now you could visualize the GQDs because they have gold on them. Another thing they used was NMR, nuclear magnetic resonance. And this is, in the simplest terms, it's essentially uh, they use magnetic resonance to see if you hit a molecule or I guess an atom at specific wavelengths, if the vibration matches with the resonance, then the atom actually like flips. And then that's really cool because once you stop hitting it with that magnetic resonance, then it unflips, and when it unflips, it releases energy. And then this energy can be utilized to the scientists can like have data about this energy and then see, like analyze what molecules or like what elements and what parts are being affected in whatever interaction they're looking at. So it's a little bit complex, but um, it's essentially just another way to visualize and localize the interactions in these very small microscopic, uh, I guess, reactions. And then another method, they used RMSD and SASA. These are just abbreviations, but what it is essentially is looking at the interactions between molecules. They were observed and they were seen using simulations. And from all of this complex methodology, it was found that the negatively charged carboxyl group of the GQDs and the positively charged portion of the A-syn were interacting. Also, it was determined that fibril dissociations were seen as a result of hydrophobic interactions. So, yeah, the main things to take away from here is that GQDs have negatively charged groups and the A-syn has positively charged groups that basically are the main parts of the interaction. And also the dissociations that I talked about are previously, are primarily from hydrophobic interactions. Okay, and then the next findings, GQDs and PFFs. So PFFs are just preformed fibrils. They're essentially just man-made fibrils. And then these are kind of the findings the researchers had in regards to GQDs and PFFs specifically. Uh, like I said, it was kind of seen that GQDs were able to kind of prevent but also reverse. And in this specific case, GQDs were able to reverse the cell damage and toxicity caused by the preformed fibrils. Um, you can also see that from this Western blot, it's just the GQDs also decrease the amount of PA-syn. So that's I forgot the exact name, the phosphorylated acin, I think. And that's essentially just what the Lewy body excretes. So they were able to decrease the PA-syn, which means that they were either able to mitigate the Lewy bodies or kind of destroy the Lewy bodies. And then in regards to neuron transmission, I feel like this visual helped me understand it a lot. So in this three compartment culture, they had three different compartments. The researchers placed GQDs in one of the three compartment cultures. They placed it in cell one, and the other one I mean culture one, and then the other one they put it in culture two. And it was seen that GQDs prevent the asyn I mean the PFF seeds from like traveling down the cultures, but it also stopped the transmission itself. So from C1, you can see there's nothing past C1, so they're preventing it from going any further. And then from the second culture, you can see that the GQDs in culture 2, they actually stop it where they are. And then findings, GQD in the blood-brain barrier. So this was also another really interesting finding that they had. They also used the biotin streptavidin hook once again, and this time they used little fluorescent quantum dots to kind of visualize everything. And they saw that there's actually 100% permeability of the GQDs through the blood-brain barrier. And this permeability they saw was kind of facilitated by the GQDs being kind of endocytosed by some cells, kind of crossing the BBB through these cells and then being kind of exocytosed or puked out on the other end. And they did in vivo mice trials. So the mice, um, they were given specific tasks or motor tasks to see how their motor skills, I guess, were impaired because the mice already had PFFs applied. So they would have their motor skills impaired, but the mice with both PFF and GQDs actually maintained their motor skills. Um, and then it was seen that the substantia nigra and the striatum were the key areas that the GQDs protected. And like I said, the substantia nigra is basically the main portion of neural damage that causes Parkinson's disease. So this relates this potential GQD therapy a lot with Parkinson's. And then one last thing I want to mention from here is that A53 human mutation was given to transgenic mice. So basically, foreign human genes were implanted into mice, and this gene was like the most common mutation for hereditary Parkinson's within humans. So when mice with this gene mutation were had GQDs, sorry, one second, had GQDs applied, they were more protected. And now discussion. I kind of labeled this discussion. It wasn't really outlined as discussion in the um, article, but I guess it kind of is a discussion. So the main thing to take away from this is that the researchers compared what they found about the GQDs compared to other potential therapies, such as nanogeos and RGQDs. And the one thing they found about GQDs that made it different, or I guess a couple things, was that the GQDs strongly dissociated the fibrils. Another thing is that the GQDs were non-toxic in living cells. These other therapies proved to be a little toxic in cells, and obviously that's not good. And finally, the GQDs had unparalleled blood-brain barrier permeability, like it was mentioned earlier. So that's obviously plus for the GQDs. And this kind of goes to show how, from this research, you can kind of infer that GQDs may be the potential number one therapy for Parkinson's disease in the future. Okay. Um, I know that was a little bit rushed. I was trying to get everything done, but it's 7:17 and we made it. So, I guess right now the main thing the best thing for us to do would be to kind of have a discussion time or ask questions with Professor Winston while she's still here. So, yeah, if you have questions, please ask.
I'm going to jump in and start off and say Parkinson's disease is definitely not my area of expertise, so I'm going to try to do the best that I can.
Hmm.
And this is also like a really novel, interesting kind of nanotherapy type of approach. It's also something that I don't do personally, but
Hmm.
I'm happy to kind of walk through and suss out anything that potentially may have been confusing. So I'm just throwing that disclaimer out there now.
All right. And
Go
also, yeah,
ahead.
you can ask any type of question, like any question
Oh
regarding
yeah, please
neuroscience.
do.
Yeah.
Yes, let's do that.
I don't want to just restrict it to this paper.
Okay, good.
All right. I'll just go down the list from my thing. Dolce, you can go first.
Okay, so my question was, can does GQD has any side effects?
Hmm.
Professor Winston, if you want to answer that, you can. I can try answering too.
Yeah, I was about to say I have would have absolutely no idea because I didn't read the paper,
Mm-hmm.
unfortunately. Um, but I guess the if I'm thinking about how I would visualize these, they look like they're like nanovesicles, right?
Mm-hmm.
And so you would be characterizing like the outside of it, and then what's being carried on the inside.
Mm-hmm.
And so I think I can imagine that anything would have some sort of potentially off-target side effect, meaning it went to a location that it wasn't supposed to go.
Mm-hmm.
That's usually some of the things that I think about when you're like delivering something to a cell. Um, but yeah, it would have to, I would probably need to look a little bit more to see if they even discuss anything about that. So that's a great question.
Mm-hmm. Yeah. As for me, in the paper, it only highlighted the side effects of the GOs and the RGQDs, not the GQDs itself, or maybe I just missed it. But
Hmm.
yeah, I'd agree with Professor Winston in that maybe if it was targeted wrong or like if it was targeted in a area that it wasn't supposed to be, then that would be probably not good. All right, Kaylee, you can
Okay.
go next.
Okay, so I have two questions and they're both like about the research too.
Okay.
So
Oh
yeah,
wait. Um,
uh,
I feel like questions regarding specifically this research we can maybe do after Professor Winston leaves. So
okay, after? Okay.
yeah, hold that thought. And then if you have any questions regarding neuroscience in
Mhm.
general or like Alzheimer's disease, because I know we're going to be doing a paper by Professor Winston pretty soon. So yeah, if you have any questions on Alzheimer's disease or neuroscience research, neuroscience in general, now's the time to ask that.
Okay, I got it. Thank you.
Mhm. Arnav, you can go.
Hello, can you guys hear me?
Hmm.
Yeah, yeah. Yeah, so my question was that I know in like Alzheimer's, I think another big protein that plays a role is tau. I
Hmm.
guess that's how you say that.
Hmm.
Um and I've like read a bit into that as well, and it seems that both tau and a sign it feels like I think they're both proteins that misfold, I guess.
Hmm.
So like I know A sign does like B sheet proteins, and I don't know how tau works, but would it be possible for a GQD to target tau protein?
I mean, I think anything's possible, right? So if you're, and I think that's a great way to look at it, right? So alpha-synuclein, or asyn is how we would call it. They're both in the category of being misfolded proteins. So alpha-synuclein is traditionally characterized for diseases like Parkinson's disease or Lewy body disease, while amyloid and tau, they're also misfolded proteins, but they have a preference for developing what most people would call Alzheimer's disease, or what we actually like to say, just kind of like this umbrella term of dementia. And so, if the idea is to target the aspects that causes these proteins to misfold, then it would suggest that if it can target the same misfolded proteins and prevent those interactions in alpha-synuclein, then in theory, it should do something very similar for amyloid and tau. The principle is makes sense, right? But these three different proteins actually take on slightly different conformations. So that's why it also may or may not be a valuable target. So with tau, for example, they actually turn into neurofibrillary tangles. So their conformation is more tangle-like versus amyloid or tau, which is like these plaque-like structures. So think of kind of like a concentrated glob of protein that just kind of twists and folds onto each other and kind of just sits into the cell body, while tau, which is usually on the outside of the neuron, that is actually what is initially known to stabilize microtubules. So you have the head of the neuron and then you have the axon. So in order to keep that in like that nice long filament structure, almost kind of like a highway, tau is on the outside of that and it kind of reinforces that. But when tau becomes phosphorylated, it falls off and then it clumps together in a slightly different conformation. And so one of kind of the basic biological kind of constructs is like structure begets function. So how the structure of the molecule, whatever form the structure of the molecule takes, is going to inform its function and what it does. And so because these proteins actually take on different shapes, sometimes things like this application may not work for all the molecules. So that was a very long and exaggerated answer. So to shorten it, it's a maybe. It's possible, but people in our field, we're really kind of specialized, right? So like I don't do really anything with Parkinson's disease, alpha-synuclein, or anything like that. I kind of stick in the AD, tau, amyloid space. And not to say there aren't people that cross over because these proteins do cross over and talk to each other all the time. But in that paper that was presented, yeah, they could literally do the exact same thing, just look at two different proteins and could possibly get a different result. So they would actually have to do it in order for me to really be able to answer that question.
Right. Yeah, makes sense because of
Mm-hmm.
like all the shapes and stuff. Yeah.
Yep.
Thank you.
Mm-hmm.
All right, Ender, you can go next.
Um, there's another type of protein issue called with PRPC, which is the prions.
Oh yeah.
Would
Mm-hmm.
I know there are some prion diseases like chronic wasting syndrome, I think it's called. It's what happens with deer, where it's transmissible through if a deer with the disease drinks water, and then another deer has the same intake. Are there any similar examples of that with the proteins that are in Alzheimer's or Parkinson's that you know of?
So yes and no. So prion, so in one wheelhouse, prion is like amyloid tau alpha synuclein. It's its own
Mhm.
entity, right? And then prion can also be looked at as a behavior. So what makes prion diseases so devastating is their ability to propagate and to move
Yeah.
throughout systems very efficiently. They're almost virus-like, if you will. And so that is the fine line between prions and amyloid and tau, because amyloid, tau, alpha-synuclein are also thought to propagate. They're also thought to move throughout the brain, and that's how they kind of affect different areas at different times. But prion, the protein itself, just can do it a little bit more quickly and at a kind of a different, they undergo just like a slightly different mechanism. And so, again, in theory, you could say yes, but they're in like it's an entity on its own, if that makes sense.
Yeah, okay. That's my favorite part to research is prion, so thank you.
Mm-hmm. Mm-hmm.
All right. Um, Syed, is that correct? Sorry.
It's Samyan, but things.
Okay. Oh,
So my
Samyam,
question is
okay.
more, it's okay. My question is more, it's not really just the neuroscience, it's more about research. In this club, we're doing just like more analysis. But since you are a professor at USC, I wanted to ask if you are open to doing research with students, whether that be virtual or in a lab,
Mm-hmm.
because I I really think that it's important to analyze research. I think that's great that we're doing that here. But it's great working with you, and I think that um, I want to ask if you were open to that, like actual research with you.
Yeah, absolutely. I take students from all over, and that's why I kind of like tried to reemphasize in the beginning, like where I'm located, because people see that I am a professor at USC, but yeah, I'm technically in San Diego. So not unless on the off chance, sometimes like people will be at school in LA, but their parents live in San Diego, and then that works out well. Or if it's something virtual, because I always have data to analyze. So I'm definitely interested in exploring those options, and in a realistic way, right? Like y'all are high school students, I have college students, and I just try to work around their schedule as best as I can, because I wouldn't probably be where I am today if I just didn't get that real world experience. Like you can't wake up and say I want to be a scientist and have never worked in a science lab, never handled research. Like you truly won't be able to capture what it's like to be in this field unless you do it. So yeah, I'm absolutely open to that.
Okay, that's great. So
Hmm.
for that, should we like email you? Should we contact you? Because I think I currently live in North Hollywood, so San Diego is quite far. But
Right.
if there is a potential like virtual research program
Hmm.
with you, or just any opportunities, should we just like email you or
Yeah, just email me and then we'll kind of set up like a little meeting to discuss like what potentially could be worked out. Yeah.
okay. Thank you.
I'll put my email in the chat too.
Awesome. So yeah, if you have any potential research ideas or if this is a great opportunity, re make sure to reach out. I know Professor Winston, you probably have to go soon, right?
Hmm.
All
Yeah,
right.
yeah, yeah, yeah. I'm just gonna be driving, so I'll probably jump back on, but you just won't see my camera. That's the only thing.
Okay.
Um, I saw Jaden saying, I don't know if you had a question for me or not before I switch over.
I mean, Jaden, do you want to ask your question?
Yeah, I was wondering if there's any new, like, I guess, recovery methods or like medications that are new for treating Alzheimer's.
For treating Alzheimer's. So, actually there are. So there are these monoclonal antibody-based therapies. And I hate myself that I can't remember the names of them. They're like Dogenimab and Mecanimab. It's an ab at the end of it. So so the answer is yes. But I will say is that it's actually been quite controversial. Just given the just given the perspective of how the clinical trials were designed and operated. And
So
with the
it's not
biggest
recommended to be used at the moment.
No, I definitely am not going to say recommended. That's kind of a strong word. It's more so accessible.
Oh,
That's kind of so when they developed these therapies, and when they in the populations in which they tested them on, they were not diverse at all. They were 99% treated and tested in a very specific population, which was not representative of just even the US population. So that was one of the biggest issues that had come out of that trial. Did they demonstrate efficacy? Meaning, was there any kind of augmentation in cognitive burden and brain pathology? The answer is yes. It was minimal, but it was a yes. And so with that being a yes, and it being one of the first clinical trials that actually had some positive data, because hundreds of trials have gone on before this and they've all been negative. Some of the therapies have actually caused harm. And so, given that this trial actually demonstrated a positive benefit, then it was rushed to FDA approval. And then, technically, yes, you could be prescribed this particular antibody if you fit a bunch of criteria. With that said, it's not accessible. The drug is ridiculously expensive, and that criteria that your loved one would have to meet in order to be able to even consider to get in line to get it is so strict and so regimented that it almost seems like it's not available or it's not applicable to all. So we still in the field have a long way to go. Cure, I absolutely not. I don't think we're there yet, but so it's an intervention. It's been shown to slow down the progression, But unfortunately, that is kind of the state that we're in, is that we don't have a cure. We've been looking at this a little too late, and we realize now that we have to identify people early, way before symptoms begin, and that's going to be the point of intervention in hopes of coming up with a cure. A lot of the clinical trials and even the drug that, is available now, in my opinion, it still was done too late. Like those patients already had symptoms, and what we have just seen is that unfortunately, once you start to fall off that hill, you can't come back from it. So at best, it's slowing down the progression, which is still revolutionary, right? If we can give you back 5, 10 years of your life, I'm pretty sure anybody would take it, but we definitely do not have a cure.
Okay, thank you.
You're welcome.
All right. Professor Winston, you can feel free to leave.
Okay.
Hop back on if you can, but yeah, we'll just be talking and having a discussion.
Okay, thank you. And
All
I
right,
will
thank you
see
so
y'all in two
much.
weeks. Thank you. Bye.
All right. I'll give y'all like, I guess, 2 minutes to try and think of any questions you have specific to this research if you don't have any yet. And then we can have like a little discussion. And then from I guess 7:50, I want to kind of ask about like how discussions should be further, I guess, how we should start doing discussions later on because some people were talking about how we might want a little bit more organization or whatever. So yeah. But I feel like giving y'all a little bit of time to think of questions might be better. So 2 minutes. All right, it's 35. So yeah. Uh Emily, you can go first. Sorry if I mispronounced that.
No, you pronounced it fine. Can you guys hear me?
Yeah.
Okay, so my question might be a little basic, but we were talking about how like GQDs could be like a pretty good like therapy in the future and stuff like that. So I was just wondering like what do you think the timeline would be of it actually being like a therapy that like everyone could use? Like do you think there would be any like difficulties or challenges? And then like do you think that they would be able to help like other neurological conditions like maybe Alzheimer's or something?
Oh, that's a great question. Um, for me personally, I would have no idea on the timeline. This was like one paper I read on GQDs. I had no previous knowledge regarding GQDs, and I didn't know they had anything to do. They had any capability of, having anything to do with neurodegenerative disorders. So, yeah, I wouldn't know any sort of timeline. This paper, obviously, um, it's acting like it might be something big, and the research suggests that it might be. So hopefully, something more things can be developed because this is also a really recent paper. I think it was from a couple months ago. So yeah, it does show a lot of promise, but I can't give you a specific timeline. And because this paper was only a couple months ago, there hasn't been a lot of research done on like GQDs versus other types of neurodegenerative disorders or misfolded proteins like amyloid or tau. So, or even prions. So I wouldn't be able to confirm for sure. But like Professor Winston said, it really depends on the protein structure, especially for this. Like you can see, that as they did like the NMR and all the visualizations, it was seen that the negatively charged portion of the GQD and the positively charged portion of the ASIN were the ones that are inactive. So those are very specific parts of the protein and the GQD. So that would probably mean that GQD wouldn't be able to function as well with, say, amyloid or tau, if you get what I mean.
Yeah, I got it. Thank you.
Um, yeah. Also, if anyone has anything to like has anything else to say, feel free to chime in or just like message me on the group, text it in the chat. Kayla, you can go next.
Okay, so my question's also kind of like about the benefits of GQD. So if you like say you don't have the Parkinson's disease, but I think one of the benefits of it was that the GQD can like reduce neuronal death. So if you don't have Parkinson's, like if you still take the GQD, will it increase your brain alertness? And is that like an independent benefit of the GQD?
That's also a great question. Um, I'd say the GQDs may when they said reversing neuronal like cell toxicity or whatever, that's implying they were reversing what the damage that was done previously by the ASINs. So to healthy brains that don't have cell damage from misfolded proteins or like ASINs particularly, I wouldn't think GQDs would have a specific reversing effect because there's nothing to reverse. They aren't really promoters for enhancing, they're more promoters for reversing the damage that's been done.
Okay, thank you. That makes more sense.
All right, Arnav.
So in the paper, it says that GQD breaks down fibrillus and like the fragments just sort of like disappear after 7 days.
hmm
But like they really don't like tell us like how the cell, I guess, handles those fragments, like
hmm
what happens to them. So my question is like what's the I guess what happens to those disaggregated fragments after GQD? Like does the cell just like get rid of them or does it escape to other cells or
hmm
yeah, or does it get degraded, I guess, is my question.
hmm So in the paper it says it disappears after 7 days. And from that, this is what so I think that means that so the fibrils were broken up into fragments, right? And these fragments after 7 days, they were so completely dissociated that they disappeared. So essentially implying that these aren't harmful whatsoever anymore. They're not even fragments, they're literally molecules just floating around. And I'm pretty sure it's like such a basic thing for me to forget, but forgot in the synapse, there's stuff that takes up the waste. Um,
Cells or
yeah, like what was it? What it
The
was
way
it
is in
was
like AP Psych. I think I
yeah, it was an
remember.
AP Psych, I know. Um, but yeah, there's like there's parts of the synapse that reuptake waste from the synapse, and there's also obviously like the cells around like is it the astrocyte? I forgot. Like astrocyte glial cells, one of them. Anyways, they filter out waste throughout the brain. So yeah, they'll probably take up the molecules from there. But yeah, after 7 days, it's basically they're literally just molecules floating around. They're not anything like not even
So it just
molecules.
gets
Like yeah, they're really small.
like yeah.
hmm
Thanks.
All right, Jaden.
I couldn't remember if the paper talks about it, but do you think like the genetic differences in individuals with Parkinson's that like affects how well or bad GQD like works?
hmm So I think GQD, the genetic differences you might be mentioning like A53. I think any genetic difference in regards to Parkinson's is more about how hereditary and how early onset it is. Once you get Parkinson's, the pathology is usually the same. It's the misfolded proteins. It's not like the genetics change the amount of like the shape of the misfolded proteins or whatever. It's just that like the genetics can predict or give you a higher risk of developing Parkinson's or give you a higher risk of developing Parkinson's earlier on. So yeah, GQD, for like if a person has the A53 mutation, then GQD might be able to might be applied earlier on in their life when they first, see signs of Parkinson's. But the genetic differences wouldn't really mean that the GQD would have a different effect on the proteins themselves because the genes don't change the protein. They just change the time that the protein like starts misfolding. Hmm. All right. Shreya.
Okay, so that process of like endocytosis and exocytosis that allows the GQDs to like transport across the blood-brain barrier and then release into the brain. How do you think this like mechanism like may affect how quickly it works therapeutically compared to how like some other therapies work, if that makes sense?
Hey, could you repeat it again? Sorry.
Okay, so the like endocytosis exocytosis process,
Hmm.
like that's like like kind of a special mechanism that like a lot of therapies don't utilize. And how do you think like that helps improve like the speed of how like quickly it works compared to like how some other therapies work?
Hmm. So, this is just me. I'm not an expert in this either, but from what I'm getting from reading the paper, it's just that GQDs are easily endocytosed by um, the cells, like BMED or whatever. They were easily taken up by it. So, I guess you could design like interventions that are easily taken up by cells so they can cross the blood-brain barrier easily, but you can't really control if the cell takes up your intervention or not. That's up to the cell. So, like endocytosis, exocytosis, it's more about the affinity to get taken up rather than you actually changing um, the cell to endocytose, if that makes sense.
Yes, yeah, that makes
Yeah.
sense.
Um, does anyone have any better knowledge on endocytosis or exocytosis before we move on? Because I genuinely don't. If not, it's fine. I'll try to do more
Okay.
research on that too. Because yeah, I was kind of like not making it up, but like very like guessing from far away. Delina, you can go next.
My question is like how can scientists like change the size of the GQDs to make sure that they only stick to the I think it's pronounced a-synuclein, something like that,
Hmm.
clumps and not like affect like the healthy brain proteins or like normal brain activity.
Hmm. So the GQDs in this paper, it says they only affect, I mean, it says that they only affect the a-synucleins. So I'm assuming that the GQDs are the specific GQDs they wouldn't really interact with other brain cells. So I don't think it has to do a lot with size. It's just about their affinity for each other. Like GQD specifically targets ASIN in this specific research.
Okay, thank you.
Hmm. Jeffrey, you can go next.
So I'm curious if we know like how this molecule is able to actually break down. Like it's not living. How is it able to break down this protein that, I mean, I don't know, how does it have like the power to do that? Like it talked about the negative charge a little bit, but like how it's able to connect to it, but I don't understand how it's able to break it down.
That's a great question, and honestly, I had that question too. This research doesn't really go into exactly the how on like why the GQD actually breaks down the ASIN. It talks about, like you said, the specific parts that bind. And what I'm gonna infer is that like the specific parts interact and like I really don't know because they don't go into it basically at all. But the research does show that it works. So there must be some reason, some way that it does break it down. And maybe it's not known yet, because this is a really recent research.
All right, thank you.
Hmm. Ender said something in the chat. It could be a similar way to how acids react with water or acid with a base. Yeah, like if you take an AP chemistry, like acid bases. And I feel like that does work a lot with a lot of like molecular interactions, specifically in um, I guess like nanotech interventions. But the paper doesn't say anything. And also, I forgot a lot about acids and bases. So. I wouldn't really know how to apply that either. Zane, you can go next.
So from what I got from the paper, how GQD is like attached to the ASIN is through the positively charged like N site, right?
Hmm.
So like what about like the regular like ASINs which are actually beneficial for yourself? Like wouldn't they potentially also interact with those since they also have a positive N site?
Uh, the the other QDs, is that what you're talking about?
No, so like the GQDs they are supposed to target like the folded ASINs, right?
Yeah.
But there's also like other ASINs which are like beneficial to or
Oh yeah, yeah, yeah.
beneficial to our brain. So
I think they
yeah,
were talking about let me go back to where I read that. So it's specifically in the fibrils or the fibrils. I still don't know what to call it, but the fibrils that the GQDs bind with the positive site. So I because the normal acyclides they don't form fibrils, you know, they're not supposed to. So I wouldn't think that there would be that positive site that the fibrils have because the fibrils are misfolded in the first place.
thank you.
Mhm. Um, okay, Kaylee, you can go next.
Okay, for this question, I don't know if it's like stupid or whatever, but I noticed like in the research paper, it said that the GQD like doesn't lose effectiveness, but on the rats, it was only tested for 6 months, and I feel like that's kind of short for like a chronic disease like Parkinson's.
Hmm.
So like, what do you think like maybe would happen like in long term? Because I feel like the body would like tend to break it down.
That's actually a good question. And is it do they test it for 6 months or does it do they test it? I thought they tested it after 6 months. Let me read it again. Sorry, let me pull it up one more time. Okay. So okay, yeah. So they do only test it for 6 months. Sorry, that took kind of long. But what I'm also getting is that, you know, mice, they have shorter lifespans. They're probably the GQD, I mean, the Parkinson's pathology occurs much at a much faster rate in mice because of their shorter lifespan. So I guess that's what they're implying. Maybe the mice just died after 6 months. Um,
Oh wait, that's true. Yeah, yeah, I
yeah.
did think about that. It's true.
But no, that is a good point to bring up because they don't go into anything more than just saying that after 180 days. So yeah.
Thank you.
All right. It's 7:50. I mean, if you have any other questions, you can let me know. But the main thing I wanted to go over at the end of the meeting today was how we should structure group discussions so it could be more a little bit more organized and could promote a little bit more interaction. Um, I feel like a couple things that people suggested me, especially on last week, last meeting's attendance form, was that we could either do like group discussions or we could do like we can have like what would you call it? I guess we could separate into breakout rooms, and then each breakout room could be led by a leader at a point for each week. So like we'd have five like say we had four breakout rooms for next week's meeting. Then like this week I'd randomly or like depending on like how much you interacted today, I'd pick out four people to be leaders for next week's group discussions in those smaller breakout rooms. And then I could just hop around and kind of add my input or like do whatever. And then we could all come together and have like a little kind of send off round robin where we just kind of all kind of say a little short comment about our thoughts on whatever paper we're going to be reading and then leave. So that means a lot of the like at least 20 minutes of discussion is going to be inside a breakout room with one of the members leading. Um, so that was one of the ideas I had. Simon, something to say?
Yeah, so I think that's a good idea. Another idea I had was specific to just getting questions answered because obviously there's a lot of questions that people have that are kind of hard to answer. You might not know the answer. I feel like we have the Discord. There could be a channel in the Discord or something like that where we could ask questions there so that we have a preparation for the meeting rather than just like having it just go down during the meeting itself. We can have a channel where we ask questions and then you can see those questions and prepare that for the actual meeting.
Hmm. That's actually a
So
really
that's
great
my
point.
suggestion.
Hmm. Yeah, we could definitely do that, and that would be great. Of even for people who can't make it to the meetings, they could upload their questions to Discord. And also, Samia, do you have any suggestions on like, wait, if we do that, how would the meeting go? Like, would I just answer the questions, or will we like take turns trying to answer questions?
So I think what we could do is if we have that like a channel or like some sort of something in the Discord where we ask those questions on like a based on words, what you could do is you could prepare answers, potential answers for those questions, and then after we do whatever we have, your slides, like your presentation we had in the beginning, you can go through that, and then after that, you can go down the list of each question and answer it, and ask for people's opinions. Or in Discord, there's actually like a threads feature
Hmm.
so you could have people make threads with their questions, and inside those threads, people can be like, answer specific, or like ask specific things or answer specific things within that thread. And
Hmm.
so we can just go based off that conversation. At least that just gives us some sort of preparation for the meeting rather than having a bunch of people raise their hands. Sometimes we might not have enough time for questions. So
Hmm.
I think that's
Hmm. Awesome. Yeah. And one thing I wanted to ask y'all, one sec, Arnav, I'm getting to. Um, one thing I wanted to ask y'all is if there was a question on Discord and if I were to ask like say a random person here to answer that question, would y'all be like confident enough to answer it? I'm not expecting like perfect answers because I personally don't know everything either. Like obviously, no one here knows everything, but would y'all be interested in like you yourself kind of answering or you yourself contributing to the presentation that I make every week, like adding little parts to it so you can I guess have a more I guess a greater contributing role to this club.
Yes.
I think the presentation idea is good.
Okay. Yeah, so I'll try to like maybe for this week, I'll try to like assign specific slides or specifically like sections to certain people or like I'll have like a Google form and then the first 5 people to sign it will get to do it. So yeah, I'll have something like that set up. Um and then for like answering, I guess if we do that, then we could lead group discussions in more like um it's like for the say um like I was a member and I did the methodology section for the next slideshow. Then I could lead the discussion for the method for in regards to the methodology for next week's meeting. Like would that work better?
I think that's pretty great. I think what you could do is have people request to be like part of your presentation. We can work on it with you, or we can just be speakers on that. So each week we can have different student speakers to just contribute to the meetings.
Hmm.
And at the very end, when we do have the questions and whatever, those specific student speakers can answer questions about their part or just
Hmm.
try to contribute to the general questions.
Yeah. All right. That sounds great. So yeah, I think the best way to go about doing that for I guess this first week, I'll be sending out a Google form in the email, and like I said, like the first 4 or 5 people to answer, I'll let them join, um, and then from then on, I'll try to like have new people join every week. So yeah, but yeah,
So
that's
I'll interject. So the other thing too is like leave a lot of time for the whole presentation, right? So like
hmm.
if you think paper, there's like an intro, methods, results, discussion and conclusion, future direction. So that inevitably could be broken up into 4 to 5 people who would take charge of that section. And then given that y'all meet every other week, if even if 5 people don't automatically show up, then like you may gotta get to like, all right, this is your week. Like I would have a sign up sheet to see how you can fill it up. So like I would say, all right, here are the sessions we're gonna meet. Then have at the top who wants to do methods, introduction, conclusions, results, that kind of thing. And then just have people just fill in a space. So you'll know you'll only have to do this for one journal club, but that'll give everybody an opportunity to present. Does that make sense?
Yeah, that makes sense.
Yeah. And then I would say carve out, if we're meeting for an hour, I would say carve out like 45 minutes for the whole shebang, right? So that means out of 45 minutes, everybody get like 5 to 6 minutes to speak. And then the last 15 minutes could be just open-ended questions, that kind of thing.
Awesome. Okay. So I'll try to set up a spreadsheet then, and then I'll have like the methods, intro, whatever.
Yeah.
Hmm.
I think that'll probably be good. I mean, yeah, I think you could either, because like obviously if you don't know what the paper is ahead of time, then you may read the method and say, hey, I actually want to do the introduction. So that could be the idea of introducing the paper early.
Hmm.
But sometimes everything's not going to work out perfectly. But I guess the goal is to get everybody to contribute. So
Hmm.
whether you're presenting the methods or doing the intro, everybody should be participating.
Hmm. Yeah, that's a great idea. So then I'll probably be like I'll probably set something up like that. And in regards to like reading the paper first, I'll send out the paper usually by the day after the meeting, like Tuesday. And then I'll send out like the spreadsheet for signing up on like Friday so that you'll have at least 3 days to skim over the paper before, the signing up comes up.
Wonderful. Yeah,
Hmm.
that makes sense.
All right. Samian.
Um, Hi. So another thing I'd like to just add on, I think the student speaker thing is great. I think that's a separate thing. Another thing that could help you, since you are kind of like the only one reading this, you do have Professor Winston and Professor Rust, but mainly you're the one taking charge and answering these questions and hosting these meetings. I'm in another neuroscientific like research internship, and what they do is they have like associates and co-associates. So they have like students helping the actual leaders and group leads. So what you like I don't know if that's something you're willing to consider. Perhaps like you have people signing up for these types of positions to help you. That gives us more contribution to the club, and that also just lessens like the burden on you, I guess, because
Hmm.
it's all falling on you. But if you have these specific positions or just like these like assistants, I guess, to help you out, I feel like
Hmm.
that would give us an opportunity and that would like ease in things for you.
Hmm. All right.
So that's just a suggestion.
Yeah, I'll send we were thinking about doing that last year, I think. I'll like try to research like how I can specifically utilize specific positions and like what positions that would be, like maybe like attendance manager or like stuff like that. Um and then yeah, that would be a great idea. So I'll probably try to send out more I'll think about that. I'll probably like make a plan and then try to send out in an email uh by tonight. No, not by tonight. By tomorrow night. But yeah, um, thank you for all your thank you for all your contributions. I feel like everything we said today was a great idea. Here, I'll get the attendance form out on the chat, and then that's basically the end of the meeting. So thank you so much, everyone.